You said very nice things. I know you said a few nice things in English now. Thank you very much. It's been a great visit. So here we are. And yesterday I did go for a run. So I went to, I guess, the west of the city, east of the city. And I did see this. And I said, oh, that's a nice monument there. And I looked at the graffiti, and there was this NIH thing there. I'm almost certain that Nuria did it, because as she just said, she enjoyed very much our work at the hospital. So all of the work that I'm going to show you now was done there. And I'll just start a little bit historically. You know, the three conditions that I started working on back in the late 90s. MEN1, cardiac complex and McCune-Albright syndrome. And almost everything we know today about the genetics of pituitary tumors, pituitary adenomas, comes from the work on these three conditions. The MEN1 syndrome was, the gene was identified in the late 90s. I was a fellow at the time, was named Menin. And you all know this disease. 40% of the patients with MEN1 get various pituitary tumors. Mostly prolactinomas, but some also corticotropinomas and other tumors. Cardiac complex is rare, although not as rare as we thought it was. I found a gene for this disease called PRK1 alpha now, and 75% of the patients with cardiac complex have abnormal growth hormone secretion, which is unique because only 10 to 15% of them end up developing a tumor. And I'll show you a slide or two about this. The process through which patients with PRK1 alpha mutations get a tumor at the end is not completely clear. I'll talk about it in a second. And of course, just like in cardiac complex, in McCune-Albright, also about 3/4 of the patients have abnormal growth hormone secretion. Just a few things about MEN1 that are relevant to what today, and I'm glad the last session before me was on precision medicine, really. First, we have some rules about, you know, when most tumors with MEN1, for MEN1 patients develop. You all know that most tumors develop in the second and third decade of life. However, pituitary tumors can start as early as the age of 3.5. So this is a tumor that I saw in a child, and to this day, he remains the youngest affected patient with a pituitary tumor in MEN1. So the screening should be individualized. And I know that we have guidelines. In fact, I'm part of the guideline writing group. We will have the new guidelines issued next year, and it will be a meeting in Marseille, in France. But you can see here the classic tumor. Probably it's better with this. The prolactin and growth hormone producing tumor that happens in these patients with a bleed inside. There's an experiment that shows basically that the entire chromosome 11 is missing. This is the centrometric probe and the probe for MEN1. It should be 4 signals, so we have only 2. So 1 long arm of chromosome 11, we should have 2 long arms of chromosome 11. This is the same patient. Yeah, it's true that I have long relationships with these patients. And this is the same patient I saw on September 19, 2018 at the clinical center of the NIH. He's doing well today. He just had his enteropancreatic tumor removed. But relevant to precision medicine, today there are more than 1,600 mutations in the MEN1 gene, and not all of them are classic mutations, either deletions of the proteins or insertions. Not all of them affect entirely the function of the gene. Most of them are mild mutations, and we haven't had the opportunity to test them in the lab to know whether they in fact affect the function of the MEN1 protein. So this is a child I saw from Chile, and you can see he had a classic prolactinoma which responded very well to cabergoline. So this mutation here was a splicing site mutation that we had the opportunity to test in the lab. Nobody else in this family had anything else except for a grandmother who also had a prolactinoma. The father, we tested him, who was the obligate carrier. He had a small lesion in the pituitary, but all the hormones were normal. And none of these patients, none of the affected members of the family had any other manifestations of MEN1. So what did this mutation do? What this mutation did was to change the distribution of the menin gene from being primarily in the nucleus, where it would act as a tumor suppressor gene, to be primarily in the cytoplasm. You can see this in this experiment where we did whole cell extracts and nuclear extracts. And we could show that the mutation changed the distribution, nuclear versus cytoplasmic. So this is a peculiar way of a mutation rendering an abolition of the tumor suppression effect. And I would argue that many of the variants that we know are there today, and you see in individual families or cases where you cannot be convinced that clinically there's MEN1, many of these variants have these weird effects, not weird, but small effects on the function of the protein. And that's how I think we have to think today when we look at whole genome screens, whether it's whole exome or whole genome, it doesn't matter. But when we look at whole genomic screens and identify a variant in a patient, I think the most important thing is to look at the patient first. And by itself, a 9-year-old with a macroprolactinoma is an extraordinarily unusual event. So I don't have to convince anybody that this patient has MEN1. He may not have the whole syndrome, but I don't have to convince many people that a 9-year-old with a macroprolactinoma with a variant in the MEN1 gene, which I had the opportunity to test in the lab, but imagine being in any hospital in the world and seeing that variant and you don't have the opportunity to test in the lab. I don't have to convince people that this is in fact MEN1 related. Okay. Acromegaly in Carney's complex, these are old slides you can see, but just a point to show again that in Carney's complex, just like in McCune-Albright, you can have for many years this hyperplasia that progresses to tumor formation. This is a 32-year-old with you can see, if you are a good clinician, you can see the prolongation of the cartilage of the ear there. You can see it, but there's really nothing else. And there's an abnormal OGDT, but clearly the patient is not acromegalic in the sense that we call acromegaly. Here's an acromegalic patient with Carney's who actually died of complications of acromegaly at the age of 65. So it takes about 30 plus years for these patients to develop tumors. Carney's complex is due to abnormalities of the cyclic AMP signaling pathway. The gene we found the first gene we found on this condition back in 2000 regulates abnormal cyclic AMP. So basically, it's the regulatory protein of the protein kinase A tetramer. When this gene is missing right here, the catalytic subunit is free to phosphorylate and targets. And if it does that without any regulation, you have tumor formation. In March of 2014, we published this one case, and PRKC beta, the catalytic subunit beta of the pKa, seems to be mutated in a defective, I should say, not mutated, because when it's mutated, it does other things. But when it's defective, it seems to affect the skeleton and pituitary tumors and also causes myxomas. So it's a form first of Carney's complex. It's not exactly Carney's complex. This patient convincingly had amplification of PRKC beta. You could see that also in the PK activity in the fibroblasts. There haven't been any other patients with PRKC beta amplifications since, but there have been patients now with mutations in PRKC beta. So it's a cause of a very rare cause of endocrine disease. Okay, McCune-Albright syndrome, we all know what these patients have. And again, the abnormality here being the GS alpha. Very uniquely, this is a proof that in some of these tumors, growth hormone can be a tumor stimulant. So for example, this is a patient that I had at the NIH, classic, I will never forget this patient. You can see the fibrous dysplasia of the skull. You can see the brain being constricted because of the fibrous dysplasia. And of course, the pituitary adenoma producing growth hormone, making the fibrous dysplasia even worse, making it basically an inoperable tumor. To make things even worse, you know that when you try to radiate this tumor, you can actually cause somatic mutations of the fibrous dysplasia. And there's a couple of cases of irradiated tumors, that fibrous dysplasia tumors, that actually became fibrosarcomas. Now, these were the three classic ones. And in 2006, there was this Finnish paper by Altona's group that published the first AIP mutations in Finnish families with growth hormone and prolactin secreting tumors. Here's a patient I saw, and Adrian Daly is here because Adrian Daly and Albert Beckers in Belgium have probably the largest cohort in the world of AIP mutants, and they can tell us whether what I'm going to say is true or not. It is my clinical impression, Adrian, that a lot of the patients that have AIP mutations and have expression of their disease before the epiphyses close tend to have somewhat of a marfanoid habitus. And this is something that I got from this first patient I had that had an AIP mutation presenting with gigantism before, in other words, the epiphysis closed. And you can see she was actually referred to me from Puerto Rico for marfans. And we found the AIP mutation. It was clearly abnormal growth hormone secretion. She was operated. And as far as I know, she's healthy today. We included here in this paper here, and I'll say more about this first cohort we studied. But if you see today in the clinic a giant, so an adolescent with excess growth hormone secretion that has a marfanoid habitus, that, in other words, the arm span is significantly larger than the height, then you have to think of AIP. That's a clinical way of picking up AIP, I think, versus other types of gigantism. AIP causes a lot of other mutations. This is from a very classic paper by Albert Beckers in Endocrine Reviews. And you can see, we have seen a few cases of Cushing's disease with AIP mutations, and I know Albert has too. The gene does a lot of interactions. It seems to be linked to phosphodiesterase. We heard today the talk earlier on the connection between AIP and RET signaling, which was published by Marta Carbonitz earlier this year. So it's an interesting gene. Clearly, I don't have the time to go more into what has been called the AIP interactome. And shortly thereafter, after the AIP was found, P27 was identified being mutated in what we call today multiple endocrine neoplasia type 4. Many of you will see it as multiple endocrine neoplasia type X. That's only in rats, not in humans. In humans, it's multiple endocrine neoplasia type 4. And what is it? It's pituitary tumors, pheochromocytomas, paragangliomas, and occasional other endocrine tumors. Primarily, in other words, the pheochromocytomas and the paragangliomas and prolactinomas. We have now found P27 mutations in sporadic prolactinomas as well as sporadic ACTH producing tumors. In this paper that we published back in 2010 with Albert and Adrian, we showed that in young patients, you can have, with Cushing's disease, you can have AIP mutations or MEN1 mutations. And to make the long story short, this is now from 2010, we started this work. We are now continuing this work from my cohort at the NIH in 2022. And this is current data that we just published in the Journal of Genetics in Medicine, which is a Nature publication. It just came out a month ago. And what we show here is that out of our 268 patients, we've had 5 MEN1, we had 2 of the AIP, of the FIPA case, but no AIP mutation. We've had 1 AIP known, 1 MEN1, 2 MEN4, so this is 2 P27. And we had 1 patient with a tuberous sclerosis. But as you can see, there's a lot of work that remains to be done. We do believe that we're now going carefully through every variant of the genes we find, and obviously it's painful, but it's work that needs to be done. And I'm doing this work with Laura Hernandez Ramirez, who once I moved out, we are still having our data at the NIH, but Laura, I'm in Greece. I go back to the NIH every couple of months. And Laura is now at Mexico City, where she's continuing this work with us. So bottom line is, the average pituitary tumor patient that doesn't have a known family history, at least based on this data, has about a 5% chance of having an inheritable genetic cause, which makes these recommendations that we first published approximately in 2012, I think, still very valid. Basically, if you have a patient that presents to you with a prolactinoma or a growth hormone producing tumor before the ages of 20-25, you have a 50% chance of identifying a mutation. If you have Cushing's disease, unless there is something suggestive, either from the clinical history or the family history, I don't recommend genetic testing at this point, despite all the work that we're doing. There are some new associations, one of them being the association with pheochromocytomas and paragangliomas in the context of succinate dehydrogenase mutations. And the main proband of this condition was this patient that I saw at the age of 27 years old, presents with acromegaly, responds to Dostinex and surgery. He's doing very well, but then during the evaluation, we found that he also had bilateral carotid tumors. And here's the two carotid tumors on the angiogram. And then, of course, when we did all the screening that we heard before from the nuclear medicine talk, you can see this patient also had bilateral pheochromocytomas and multiple paragangliomas. His family, we took out the tumor. And so the question is, what mutation did he have? He had the succinate dehydrogenase D mutation. And the question is, of course, at the time, that nothing was known about succinate dehydrogenase variants causing pituitary tumors. The question was whether SDHC would cause a pituitary tumor. You can see that in a normal pituitary, there's plenty of SDHC expression, but when you do Western blot or when you do the immunohistochemistry in the patient's tumor, so here's the Western blot, normal pituitary expresses SDHD, but the patient's tumor doesn't express SDHD, and of course the immunohistochemistry doesn't. So when we do the allelic heterozygosity, there's clear loss of the allelic of the normal allele. So very clearly, there is a complete loss of the SDHD locus on this particular patient. So suggesting that perhaps this variant is causing pituitary tumors. We collaborated with Marta Karbonitz and tested her cohort of patients with pituitary tumors and pheochromocytomas and paragangliomas. So to make the long story short, every time you have in a patient with pituitary tumor a family history positive for paraganglioma and pheochromocytoma, you have an approximately 75 to 80% chance of identifying a succinate dehydrogenase variant in any of the 4 subunits: SDHA, SDHB, SDHC, and SDHD. We named this condition the 3P association, pituitary pheochromocytoma and paraganglioma. Here's another patient. So the previous one is a prolactinoma, and here is a non-functioning pituitary tumor. One thing we noted, and I have to give credit here to Marta Karbonitz for this, because she asked me, Konstantin, have you seen this in many of your tumors? And I told her, yes, I have, but I had not reported on it. So Marta and I went back to look at all the slides of the tumors that had succinate dehydrogenase mutations. And we found out that most of the tumors, including now in the mouse, this is the mouse pituitary of succinate dehydrogenase B heterozygote mouse, had these vacuoles within the cells. And what are these vacuoles? Well, we did electron microscopy in the hyperplasia of the mouse, and you can see these vacuoles are basically unhealthy, destroyed mitochondria. This is a very long mitochondrion, so unhealthy. And then here, a mitochondrion that's basically broken down. So this is debris, unhealthy mitochondria that you can see. So today, if you have a pituitary tumor and the histologist reports a vacuole like this in the mouse or like this in the human, then that's a hint to test for succinate dehydrogenase, because I don't think there's, there's a pathologist. A pathologist was online, and he's not here with us today. I hope he's listening, but there's really not a lot of pituitary tumors that have this vacuole. So I think this is pathognomonic for that. Obviously, when you have patients, now it's very important when we see in the clinic patients with pituitary tumors to ask a very comprehensive family history. In many of the succinate dehydrogenase variants, but also the P27 variants and the MEN1 variants, not so much the MEN1, for sure the MEN4, the P27, and for sure the succinate dehydrogenase. You can have generations of the family skipped. So you really have to go back, to, if you have the data, to 3 generations and extensive family members, because you can miss a family association. For example, it wasn't until this group in Holland looked at extensive family history of patients with variants of the succinate dehydrogenase that they picked up that they also have other neuroendocrine tumors. And this is the paper that reported this. And also, this is the paper from Australia. I'm sorry, this is also, yeah, Australia and Holland. And this showed the association of succinate dehydrogenase with renal cancer. But again, that was only picked up when 2 or 3 generations of families with succinate dehydrogenase variants were picked up. Because you see, the penetration of the disease is about 40%. And it's about 40% for pheochromocytoma and paraganglioma, and about 20 to 30% for all the other tumors we're talking about. So you really have to look. AIP is a good example of that too. In AIP, you don't have any other tumors. But here you have the opportunity to catch a succinate dehydrogenase variant by looking at a number of tumors in the extended family history. And Adrian, in 2018, Dr. Daly and Dr. Beckers published this association of pheochromocytomas and pituitary adenomas with MAX, the gene, one of the 12 or so genes now for pheochromocytomas. So the 3P association, pituitary, pheochromocytoma, paraganglioma, is not only due to succinate dehydrogenase variants, but it's also due to MAX variants. And there's a very nice paper just came out. I didn't have the time to put the slides here. Just came out in endocrine-related cancer by Marta Carbonitz, where she's talking about the overall MAX variant associated tumors, including, of course, the pituitary tumor. So the world of variants, genetic variants, germline, and I'm talking about germline genetic variants, is expanding. A few words about the work that we've done together with Adrian Daly and Albert Berkers. It all started when I saw this family back in 1998 from New Zealand. It was published in 2011 as just a case report of inherited gigantism. This boy here was 5 years old when this picture was taken. This boy was 2 years old. You can see the father here. So the 5-year-old is like just one head down from the father, right? The mom was operated when she was 18 with gigantism, which is obviously much milder. So now you have 2 boys and a mildly affected mom. And for every geneticist, this is an X-linked disease, right? So we have a mom that has 2 X's. The X, if the X has the gene, she's protected because she has a healthy X. If she passes on that disease to her 2 boys, and each of the 2 boys gets the affected X, the boy doesn't have another X, so he will be more severely affected. And it is true that most giants in the history were boys. In fact, the very first brothers, the Hugo brothers, reported by Pierre Marie in what was called at the time Marie's disease, were also boys. And so there was very good evidence in the literature and my own experience with this one family that the disease, early onset gigantism, is in fact an X-linked disease. And that ended up being true. We collected together with Dr. Beckers and Adrian Daly all the cases that had early onset gigantism. What's this manifested by? A very simple thing. So again, you have to be a good pediatrician, really, not even an endocrinologist. All you need to know to pick up these patients is giants that approximately tripled, 2.5 to 3 times the original length at birth. within the first year of life. And that's what you can see here in the growth charts. Both boys and girls tripled their size within the first year of life. And so these patients ended up having an abnormality that was de novo. In most patients or most families, it was de novo. In some cases were isolated cases, but they all had this amplification of chromosome X, that you can see here. In some cases that were inherited, it's segregated with a disease, of course, and basically it is a known, not very well known, but a known non-recurrent rearrangement on chromosome X that has been seen in other genomic disorders. The paper came out back then in 2014, and it affects a single gene, GPR101, that seems to be the cause of this extra growth that we called X-linked acrogigantism. The patients here have also hyperplasia, primarily, which then becomes a tumor. And you can see that, just like the slide I showed you before in Carney's complex, you can see this progression from hyperplasia to tumor formation with the classic reticulin stain, disintegration, and so forth. GPR101 is an interesting gene. Adrian and I and my former fellow, who was the first author in that paper, Gianpaolo Trivolin, who is now in Milan, we're all working still on GPR101. But it's an amazing gene. It's expressed primarily in fetal pituitary. It's also expressed in the hypothalamus. In the adult, it's expressed primarily in the hypothalamus, not in the pituitary, unless you get one of these tumors that produce growth hormone in the context of XLAG. So it's a developmentally regulated gene that regulates from the hypothalamus the expression of growth hormone by the pituitary gland, except for the fetal time when it's expressed throughout, and also for a probably very brief time during adolescence. So this is what this says. GPR1 in the pituitary is regulated in a cell type and developmental stage-specific manner in different species. These differences might reflect the very different growth, development, and maturation patterns among species. We were able to show in fish, very briefly, this paper was published almost 2 years ago now, that when you knock out GPR1 in fish, you do have abnormal growth. So clearly, at least in fish, just like in humans, GPR1 regulates growth. And there's a lot of elegant experiments also that Dr. Adrian Daly has done that I don't have the time to show you here. But this talk is clinical and genetic, so I want to tire you with laboratory data. The point is that in acromegaly, more than all other pituitary tumors, so in growth hormone producing tumors, more than in all other pituitary tumors, there is genetic associations, and there is genetic associations to be identified. And I always bring up this case of a man that I picked up with acromegaly, and I picked him up in my genetic cancer clinic. He came to see me for his familial testicular tumor. So he had germ cell tumors, testicular tumors, and he was coming from a family that his son also had testicular tumors, but his mother and great uncle had renal cancer. So, and then I see him in front of me and I pick up this acromegalic features, and sure enough, he had a growth hormone producing tumor. So we have sequenced everything in his genome, both the genome of the pituitary tumor as well as the genome of his renal cancer, and we haven't found anything that's consistent that seems to point what the cause of the association is here. We've sequenced everybody in his family. So there are discoveries to be made, and I'm telling you again that they are far more common in acromegaly. There's something about this particular cell lineage, the growth hormone cell lineage, that I think is more frequently due to germline variants. All right, a few words about Cushing's syndrome. Obviously, you've seen this that I've published before, and it has a lot of new genes in there. It's amazing that this table that I made, I don't know, 30 years ago, I keep putting new genes up there. I already mentioned AIP, MEN4, that's also true for corticotropinomas. I'll say a few words now about some of the newer associations here, like USP8 and DICER1. I don't have anything on some of the other USPs that have been linked now, or the CD23. So USP8 is a very interesting gene because it regulates EGFR, and work that had been done by Shlomo Melman had already shown that EGFR signaling, unlike other pituitary tumors, we'll see whether AIP links to EGFR. It's funny that AIP links to RET. So I wouldn't be surprised if AIP links to EGFR as well. But unique to ACTH producing tumors, EGFR signaling seems to be important in this type of a tumor. So it wasn't a big surprise that USP8 was found to be mutated in Cushing's disease. What was a big surprise was the frequency with which it was found mutated. Half of all ACTH producing tumors have these hotspot mutations at the part of the USP8 gene that binds to EGFR, and that's what's shown here. As you know, there's now a couple of trials on EGFR inhibitors in uncured Cushing's disease patients, and they don't seem to be very effective, and I'm not sure why. We'll see what the data show when they're published. But I'll show you here that as we see patients and we see variants, this is a patient that came to us at the age of 14. I think, Nuria, you had already left the lab when we saw this patient just 3 years ago. Clearly recurrent, classic Cushing's, recurrent Cushing's. You can see here the big tumor. Here is the patient. She had developmental delay, multiple areas of skin lesions and nail lesions, impressive hypertrichosis, which you could not explain simply from Cushing disease, and then skin patches that were pigmented. A number of other things: dilated cardiomyopathy, chronic lung disease, chronic kidney disease, and the bottom line is she had the USP8 hotspot mutation in the germline, and clearly was present in the tumor, of course. And this patient ended up dying from this disease that she had. So it's a new phenotype. I know now of 2 additional patients that haven't been reported in the publication, but the clinicians have been contacting me, and one of them in Boston at Boston Children's. So there's more patients with the USP8 variant. So this particular mutation, of course, it's one of the classic hotspots. So this variant does affect clearly the EGFR signaling, and at the time we discovered the patient, it had already been reported as a somatic change in 2 corticotropinomas. Now, carnate complex and Cushing's, you know that carnate complex is associated with primary pigmented nodular adrenocortical disease, PPNAD. But so it was a big surprise to me to find a PRK1 alpha mutation causing also not only growth hormone producing tumors, but also ACTH producing tumors. So, and how did we find this out? Well, a patient that we saw with Cushing disease who we operated the pituitary gland. He had a big tumor, so he had to have hypofisectomy. And then he comes 2 years later with classic ACTH producing tumor. He comes 3 years later with, this time, ACTH independent Cushing's. We operate on him and he has PPNAD. So then we sequenced the PRK1 alpha. And of course, we went back to the pituitary tumor and discovered that there was classic LOH at the PRK1 alpha locus at 17q. So this was the first case of an ACTH producing tumor in carnase complex, PRK1 alpha causing basically double Cushing's, both in the pituitary and in the adrenal. You could argue, maybe you can say the ACTH, the chronic excess of ACTH stimulated the PPNAD perhaps, but we know that PPNAD is unresponsive to ACTH. So I don't think it is the chronic ACTH stimulation that led to the adrenal tumor. I think it's just that we have 2 independent effects of the same mutation. I can't be certain, but I'm just saying. There are 2 other cases that have been reported since, one that was reported previously, and then one that is reported by Marta Carboni's group. So it's a rare association, but a true association, the PRK1 alpha causing pituitary tumors as well. And finishing up with a couple of interesting genes, or more than a couple. DICER1 is interesting, but only causes pituitary blastomas. It first was reported in this male infant, 9 months of age. This is a slide that Sherry Deal gave me, the physician in Montreal who saw this patient. Here's the patient. You can see the tumor. This is not an adenoma. This is a pituitary blastoma. And these are always ACTH producing. I don't know if they produce anything else. How do you pick up these patients? This is an interesting story now. Everybody who is a black dot here doesn't have a pituitary tumor. Is in the family of the patient that have all of them have the DICER1 mutation. What do they have? Multinodular goiter. So the way you pick up, so again, the importance of being a good clinician and never throwing away anything in the family history. If you have a family where you have multiple goiters and the occasional ovarian or testicular tumor in the family, that's a DICER1. That's a DICER1. So here's what DICER1 does. The pulmonary blastomas, kidney blastomas, but importantly for us, for endocrinologists, ovarian and testicular tumors. And of course, you will see this very rarely, but it does exist. We reported this variant. Nuria is one of the co-authors in this paper. So this is work that we did when Nuria was in the lab. We now know many cables-1 variants that seem to be pathogenic. What does cable-1 do? It basically regulates the response of the pituitary and the hypothalamus to glucocorticoids. So it affects the glucocorticoid receptor responsiveness in this negative feedback loop. Patients that have cable-1 variants have an inability to respond to this negative feedback, and that drives the ACTH levels, and that's how they get ACTH producing tumors. These are this is another gene that we are now putting the publication together called killin. This is on chromosome 10. It regulates PTEN expression, and there is some data that demonstrate that PTEN, the tumor suppressor, is also involved in pituitary tumorigenesis through AKT signaling. So one of the connection between RET signaling and AIP is interesting because it implicates again AKT, which AKT signaling is involved, I think, in all the pituitary cell lineages. And so we have to, when we look now at genomic data, I look at pathways and variants, and obviously machine learning will do that much better than me, but that's how I do the work right now. Right. So I think I told you some, I touched on most of these genes. We talked about AIP, XLAG. We talked about MEN1. I didn't show in slides, but we talked about MEN2, the few cases that have been reported with RET mutations. P27, that's P27, MEN4. Told you about carnate complex in the PRK1 alpha, the PRKC beta, and a locus that hasn't been elucidated yet on chromosome 2, which happened to be the first locus. And I do believe there's something there, but it's a chromosomal variant. It's not a gene, at least as far as I know. The succinate dehydrogenase, we should add here MAX. The NF1, I didn't say much about it, but there's a few cases of NF1 we published now, just this year, earlier this year, I think it was. The paper on NF1 and growth hormone excess. I told you about DICER1. I told you the couple of cases of tuberous sclerosis and pituitary tumors. Somatic, we touched, obviously mentioned GS alpha, but we touched on USP8. I did not have the time to go over PIK3C alpha and the evidence for mosaicism for GPR1 and GS alpha. And again, from the Genetics in Medicine paper, now I've included here, this is all Cushing's disease patients. I've included some of the older cases, but now the new genes as well. So the cables one, the killin variants, one germline USP8, and now we have 2 tuberous sclerosis. And so we went up in the last 5 years, we went up from 5%, 4.9% to 7.5%. And so still a lot of work to do. Summary, beyond, so it all started 20 years ago with MEN1, carnate complex, and McKean Albright. We're now adding the various Ps, P27, P18, P15. Remember what I just told you, the mild variants of all these genes, including the MEN1, but also, P27, P18. There's plenty of variants out there. One of the patients with P27 that came to us with an ACTH producing tumor, you know how you could pick up in their family history that there was something? Basically, there were several cases of colon cancer at a young age, ended up being all P27 positive. Told you about AIP. It's difficult to pick AIP because it doesn't have any other associations, any other tumoral associations. So unless you do extensive family history and you pick up other pituitary tumors, that's how you pick it up. Succinate dehydrogenase, I told you about the association with pheochromocytomas and paragangliomas, and of course the MACs. And here in the succinate dehydrogenase, you can pick up renal cancer, you can pick up neuroendocrine tumors as well. We talked about XLAC and GPR1, talked about USP8, DICER1, Cables1, and other genes. And so thank you so much for your attention. It's really been a wonderful meeting. Congratulations, Nuria, for setting this up. And thank you for inviting me to be here. Thank you.